Okay guys, welcome back to part 3 of our video on antibody production and vaccination. Today we're going to be looking at a few different things, not too many, anything too heavy. We're going to be looking at the effect of histamines and why they're produced. We're also going to be looking at monoclonal antibodies and the production of these and use within industry. And that's kind of going to bring us up to the end of this unit. So first of all, let's talk about histamines and how they can, why we have them and where they are. So in our body, we have a group of cells known as mast cells. And they're immune cells that we generally find in connective tissue like skin and muscle. And these cells will secrete a substance called histamine in response to any infection. So if we have an infection in our body, these cells simply produce this substance called histamine. Now there is another type of cell in the body which produces histamine, and they are called basophils. And they're a type of white blood cell which circulate within the circulatory system. And they also will release histamine in response to an infection. Now, what histamine generally tends to do is it causes dilation of small blood vessels in any infected area. So the vessels become very leaky. What that means is that the vessels have an increase of fluid to them, and that fluid can contain large immune components such as antibodies that have been produced in response to the pathogen. Okay, so it's a way, actually, the release of histamine is completely necessary. It's not a bad thing, okay, but it can cause us to become symptomatic, to have some pretty nasty symptoms, okay, and those symptoms really, you can see this one is the rash, this histamine rash, which is caused obviously by blood vessels leaking into that tissue. Okay, and blood is being released into this tissue. So it's not a particularly nice thing to look at. It can be itchy, it can be rough. Also, you can start to sneeze. If it's an allergic reaction to pollen, we'll start to sneeze. We'll start to have an excess mucus buildup in our throat. And in very severe cases, we can go into anaphylaxis, which is where our throat starts to close up because of swelling in the throat, and we can start to suffocate. Now, that can be obviously deadly if it's not treated or if it's a very serious case. But the good news is it can be treated relatively easily, and we can take antihistamines as a way of doing that. Very recently, I got something called scombri poisoning, and it was due to eating fish on Nusa Lembongan that had a bacteria in which had a very, very high amount of bacteria contained a lot of histamine, and when that histamine came into my body, okay, even though it wasn't the pathogen that was causing, okay, it's these cells that were these bacterial cells that were actually already rich in histamine. It caused this allergic reaction, and I experienced all of those symptoms we just talked about: a rash. Okay, I also went into slight anaphylaxis when my throat was closing up. It also felt really strange. I had this kind of burning sensation in the back of my mouth. It felt like when you've eaten something really hot and your mouth starts to it feels like you've burnt the top of your mouth and like all the skin's hanging off your mouth, like when you eat a pizza slice and you just pull off the top bit and it burns your mouth. It was like that, but it actually hadn't happened. However, I took antihistamines, and within a couple of hours, the symptoms had abated quite nicely. Okay, moving on from here. In the next section of this, we're going to be looking at monoclonal antibodies and their use. So basically, monoclonal antibodies are highly specific, purified antibodies that are produced by a clone of cells and are derived from a single cell, and they recognize only one type of antigen. Now, to produce the clone of cells that we need, what happens generally is that the antigen, that is recognized by a particular antibody is injected into a mouse or another type of organism, okay? It presents a challenge, and of course, like any other type of organism, there is a response by the mouse's immune system, okay? And so in response to that, antibodies will be created that are specific to the antigen that was injected, okay? And plasma cells will start producing antibodies, or B cells become activated, they mature into plasma cells, and the plasma cells produce the antibodies that we want. The plasma cells are then removed from the spleen of the mouse, And basically there will be many different types of antibodies during that that will be extracted from that spleen. Okay, so there will not be, some of them will be the ones you want, some of them will be the ones you don't want. You can see here those plasma cells, they've got yellow ones, red ones, yellow ones, blue ones and purple ones. Now the beta cells are then fused, okay, with cancer cells, okay, called myeloma cells. Okay, these are our cancerous cells, and obviously we know about cancers that they divide rapidly without control, and in this case that is exactly what we want them to do. We want them to divide rapidly. Okay, so alternatively to this, they, you've heard about the use of bacteria. Bacteria do the same thing. They divide rapidly, and they can be used for other purposes. But in monoclonal antibodies, they use myeloma cells, which are a special type of cancer cell. So the cells are fused together, which is what you see here. Okay, and then they're cultured to produce lots and lots and lots of those different types of cell. What happens then when they need to identify the ones that specifically exhibit the antibody that we want? They use a dye that reacts with that antibody, okay, to show up the ones, and then those are cultured to produce more and more and more and more of them. All right. Now what you can see in this next image is a manufacturing plant for monoclonal antibodies. Now these are just fermenters, just like in the same way that we would ferment bacterial cells. We give them food source, you provide them with oxygen, okay, and you get a runaway manufacture of those particular cells. So in this case, in these huge vats here, we're producing huge amounts of monoclonal antibodies, okay, that are attached to the myeloma cells. Now, what are monoclonal antibodies used for? They're used in the treatment and diagnosis of diseases. So examples include malaria or the test for malaria. And they can be used to identify whether either humans or mosquitoes are infected with that malarial parasite. The test for the HIV pathogen or the creation of antibodies for injection also into rabies victims is also done using monoclonal antibodies. The last thing we're going to look at in this is one particular kind of way in which it's being utilized is in pregnancy tests and how that works. So the way they work in pregnancy tests is, this is a pregnancy test strip, just to give you an idea. We have the monoclonal antibody here, which is attached to point C. This is this first area here, and that's denoted by these purple guys, okay? We also have a monoclonal antibody, another similar type of monoclonal antibody here, which is attached to a dye, and that is at point B, so at this point of the strip, okay? Towards the bottom, we can also see here you've got another sort of type of receptor. So in this, in urine, there is a hormone called HCG. Okay, and this is, the HCG is called human chorionic gonadotrophin. It's uniquely produced during pregnancy by the developing embryo and later by the placenta. And basically urine has quite detectable levels of HCG. Basically then what happens is urine is applied to one end of the strip. Okay, so urine, here you can see in this one, this urine contains levels of HCG. The HCG is picked up at point B by an antibody that has a dye on it. The antibody then drifts down the strip to point C. There is another antibody which attaches, and this is where the dye starts to come out, so you would get a line being produced, okay? Down at point D, there are immobilized antibodies that bind to the dye-bearing antibody down here. So if you are not pregnant, okay, generally what happens is the urine will come down, there's no HCG, okay, so the dye bearing antibody does not pick up the HCG, okay, and then obviously does not bind to point C, and just simply carries on down 2-point D and binds with the antibody there, okay? And what you should always get, even if you are not pregnant, is a line coming out at the bottom, okay? Irregardless of whether you're pregnant or not, okay? That simply means that the actual test device itself is working, all right? So if there is a pregnancy test taken, it doesn't show a line, a purple line or a blue line there, it means the pregnancy test is broken and it should be repeated. All right. Obviously, in a positive test, what will happen is the HCG drifts down, attaches to the antibody that contains the dye. That drifts further down the pregnancy test until it hits this immobilized antibody. It combines with that, and we get a purple image, the purple line coming out. So 2 purple lines or 2 blue lines generally denote a positive test, which basically means you're pregnant. I think most of them are around 97% accurate. Okay. Now there's a question which goes along with this, which is on page 475 of your book. Please make sure you have a go at it. All right. Thanks for listening, and see you later.